Hello, my name is Catherine Correro Padilla. I'm a grad student from the University of Puerto Rico Medical Science Campus. I am doing research at the Institute of Neurobiology with Dr. Cristina Velasquez, and today I will be presenting my project titled Lead-Induced Hippocampal Changes in BDNF Gene Expression and Neuronal Survival. As a matter of introduction, I will start with lead, which is a metal very well used in the manufacturing of pipes, lead-based paints, ceramic clays, battery, pottery, and ammunition. It can enter the human body through the food, water, and air, causing negative effects on the human health, and it has been proved that developmental exposure to lead causes deficit in cognitive functions, IQ, behavioral effects, and attention deficit hyperactivity disorder. There are 4 key factors of lead reported by the Agency for Toxic Substances and Diseases Registry in 2017. And the first one is that once in the bloodstream, lead is primarily distributed among 3 compartments, which are blood, mineralizing tissue, and soft tissue. We can find it more in the bones and teeth of adults, which contain more than 95% of total lead in the body. The second factor is that in time of stress, particularly pregnancy and lactation, the body can mobilize lead store, thereby increasing the level of lead in the blood. The third factor is that the half-life of lead in the adult human blood has been estimated as 28 days. And the fourth factor is that the body accumulates lead over a lifetime and normally releases it very slowly. The reason of why this experiment is very important is because according to data reported by the Iceland Water System between January 2015 and March 2018, 97% of Puerto Rico population is served by a local drinking water system with at least one recent violation of the Safe Drinking Water Act's lead and copper testing requirements. That is far higher than any U.S. state. So for the maximum concentration approved by the EPA in Puerto Rico, there is 15 parts per billion. But here in Puerto Rico in 2015, reported by Colon Ortiz, there was found a highest concentration of about 14 parts per million. So for my experiment, I will be using 2 different concentrations. The first one will be 27 ppm, which is the low-level human equivalent gestational lead exposure in mice according to Leisure in 2008. And the second concentration will be 5 ppm, which also have been proved to have cause changes in the brain. For my experiment, I have 2 proteins of interest, and the first one is brain-derived neurotrophic factor, or BDNF, which is a central mediator of neuroplasticity that promotes the survival of nerve cells by playing a role in the growth, maturation, and maintenance of these cells. BDNF is expressed throughout the central nervous system, although particularly high BDNF expression is found in the hippocampus and cerebral cortex, and is primarily expressed in neurons. And for my experiment, I will be focusing on the hippocampus area of the brain. So my second protein of interest is the receptor of BDNF, which is called TrkB. And here on the right, we can see the plasma membrane, where is the TrkB receptor, and once BDNF attached to that receptor, it will be activated, and it will promote a signal cascade, and depending on the pathway that it activated, it will promote the long-term potentiation, the synaptic plasticity, neurogenesis, survival, differentiation, or the inflammatory suppression. For my experiment, the hypothesis is that changes in BDNF gene expression are key in determining known changes in neuronal survival due to physiologically relevant concentrations of lead exposure. My first aim is to determine the effects of different concentrations of lead exposure on hippocampal neuronal survival in adolescent female and male mice, and my second aim is to determine the transcriptional regulation and gene expression of BDNF in response to lead exposure within hippocampal neurons. This diagram shows the experiment that I proposed, but because of this unusual year, I couldn't do it. But as a brief summary, I will have my control group and 3 experimental groups. In my control group, I will not be exposing the mice to any lead at any time point. The POPs will be divided in 3 different groups. The first group of the POPs will be the control that will not be exposed to lead. The second group of the POPs will be exposed to 5 ppm of lead in the water, and the third group will be exposed to 27 ppm of lead in the water. My first experimental group will be a female mice that will not be exposed to lead neither on the breeding process, but in the pregnancy process it will be exposed to 27 ppm of lead in the water, and later on the POPs will be divided, as I previously discussed that the POPs will be divided in a control group. My second experimental group will be a female mice that will be exposed to 27 ppm of lead in the water, but not anymore, not in the breeding process, not in the pregnancy process, and later on the POPs will be divided in 3 respective groups. For my third and final experimental group, I will have the female mice that will be consuming lead at every time point before the breeding process, during the breeding process, and during pregnancy. And later on, the POPs will be divided in the 3 previously discussed groups. As a proof of principle, I do immunocytochemistry with the human embryonic kidney cells, which are the HEC293 cells, with the BJ fibroblast cells and the postnatal mouse hippocampal neuron. I treated them in 3 different groups: the control, which is not exposed to lead, the second group, which is 5 ppm, and the third group, which is 27 ppm. And I exposed to no lead or the different concentrations of lead for 24 hours. Later on, I analyzed it, and the results, at least for my first aim, which is the cell survival in response to lead exposure, I have this graphical representation of fibroblast BJ cells and HEC293 cells. And in this graphical representation, on the y-axis, I have the number of cells as percent control, and in the x-axis, I have the different concentrations of lead. And what it shows you is at least that with 5 fibroblast BJ cells, with an increase in the concentration of lead, there is a decrease in the number of cells. But with HEC293 cells, something different happened, because we can see a significant decrease in the number of cells with 5 ppm of lead. But with 27 ppm, something could happen between the cells and it could rearrange or anything else, because with that high concentration of lead, which is 27 ppm, there are more cells when compared to 5 ppm. For my second aim, which is the transcriptional regulation and gene expression of BDNF in response to lead exposure, I have the images of immunocytochemistry on the left, and on the right I have the graphical representations of those images. In the images, in the first column, I have TAPI, which is the nucleus of the cells. In the second column, I have phalloidin, which is the cytoskeleton of the cell. In the third column, I will have my proteins of interest, which could be BDNF or TrkB. And in the fourth column, I have the merge of them, which is the merge of DAPI, phalloidin, and BDNF, or DAPI, phalloidin, and TrkB. For the first row of these images, we will have the control group, that is no exposure to lead. The second row will be 5 ppm of lead, and the third row will be 27 ppm of lead. For this particular slide, we have the results of BJ cells, and on letter B, which is the graphical representation of letter A, we have on the y-axis the average intensity normalized to control, and in the x-axis the 2 different concentrations of lead. And we can see a definite decrease in the intensity or in the fluorescence of those cells when we are talking about BDNF, and we also see that decrease of fluorescence with the TrkB receptor. And this includes the nuclear and non-nuclear transcription and gene expression of these proteins. For HEC293 cells, I also have the images of immunocytochemistry on the left with letter A and C, And on letter B and D, the graphical representations, and we see a different accommodation or a different image that we can compare with BJ cells, because in this case with hectonite 3 cells we see an increase with the fluorescence, and that increase include the BDNF, which is the protein, and the tract B, which is the receptor of the protein. And we see that increase of fluorescence and intensity in the nuclear and also in the non-nuclear transcription and gene expression of those proteins. With the hippocampal neurons, here we analyze the control group and the 27 ppm lead exposure, not the 5 ppm. So we have the images on letter A and C, and on letter B and D, again, the graphical representation of those images. With letter B, which is the graphical representation of letter A, we have a very marked decrease, a significant decrease of BDNF, of non-nuclear BDNF, and not significant changes in the nuclear BDNF. But when we go to tract B, which is in letter D, we see an increase in the nuclear tract B, but a decrease in the non-nuclear tract B. So for conclusion, first on BJ cells, the decrease on BDNF on tract B is showing a possible direct effect to the pathway of the proteins, blocking the possibility of the cell to survive by the exposure of lead. On hectonite 3 cells, the lead exposure causes the cells to react, increasing BDNF on tract B, which also will increase the possibility of survival after the threat. And on hippocampal neurons, the exposure of lead produces a decrease of BDNF protein and an increase of tract B receptor. There is a possibility that neurons fighting to survive the threat of lead secrete BDNF to find and activate tract B, which is increasing around the cell. For the results results have shown that the effect of lead exposure are present even in small concentrations, but react different depending on the cell type. And for future experiments, we will study the transgenerational and longitudinal effect of early lead exposure in mice, and we will be addressing directly the possibility of lead-induced BDNF abnormal transcriptional regulation within the hippocampus, leading to deficiencies in neuronal survival. Thank you for your attention, and if you have any question, I will be happy to answer them. Thank you so much.